Hey everyone, welcome to my YouTube channel. This is Stephen Kapilitan and this is Microbial Genetics. As we go and review microbial genetics from the words itself, genetics, is a study of inheritance and some of the inheritable traits which are expressed in the organism's genetic material. And with this, geneticists study many aspects of inheritance, including the physical structure, the function of these genetic materials, the mutations, the transfer of genetic materials among other organisms. And in this particular presentation, we will examine these topics as it applies to microorganisms. The study by which forming the basis of our understanding in human, animal, and plant genetics. So we take a look at microbial genetics. But before we go further with our discussion, let us start with a case study. It's entitled Another Stroke. Let me present you a patient who is Ben Swanson, a 68-year-old man, which is returning to his nursing home after spending 2 weeks in the hospital recovering from a stroke. His doctor is impressed with his progress and thinks that by continuing to work with a physical therapist and rehabilitation nurse, Ben will be able to function as independently as he did before the stroke. Ben is so excited to get back home. He misses his friends, especially his roommate Chester. But he remembers the doctor's orders. He's supposed to take it easy and rest, particularly on his first few days back at home. Ben follows the doctor's instructions. His physical therapist and rehab nurse visit him every day, and he and Chester spend time just chatting or playing cards and dominoes. On Ben's third day home, Chester decides to surprise him by bringing some ice cream from the dining room back to their room as a treat. Chester is shocked when he opens the door to their room and finds Ben lying on the floor. Ben doesn't respond when Chester calls his name. Chester quickly hits the emergency call button located in every room. The nurses and doctors arrive, and Chester is so certain that Ben has suffered another stroke. Well, to better guide us in understanding that case study, we need to further answer these guide questions. Do you think Ben has suffered another stroke? Or could there be another explanation for his condition? You may drop down your answers in the comment sections below. So this is the structure and the replication of genomes. In this particular presentation, we will study the genomes of your prokaryotes and eukaryotes. We compare and contrast their genomes. But before we do compare and contrast their genomes, let's understand what a genome is. A genome of a cell or a virus is the entire genetic complement, including both its genes, specific sequences of nucleotides that code for RNA or polypeptide molecules, and nucleotide sequence that connect genes to one another. And the remainder of this presentation will focus on bacterial genomes, their structure, replication, how they function, mutation, repair, and how they compare and contrast with the eukaryotic genomes and with the genomes of some of the archaeans. With this, we start and begin our lesson with examining the structure of the nucleic acids. Here, we will describe the structure of your DNA, discuss how it facilitates the ability of DNA to act as the genetic material. Deoxyribonucleic acid is formed in this particular figure. These are the structures of your nucleic acids. Remember, nucleic acids, these are polymers of nucleotides which are consisting of nucleosides. Okay, these are your nucleosides, highlighted in maroon for A and B, and in letter C it's highlighted in blue-green. Okay, a nucleoside is a pentose of a sugar and a nitrogenous base. They are bound to a phosphate. Okay, in letter A, we see here a base pairing between the complementary bases of your adenine and thymine. These are formed together with 2 hydrogen bonds. Okay, these are only found in DNA. These 2 hydrogen bonds you see here in this first figure, where the 80 bonds. For letter B, this is an AU base pair which are found in RNA. This is adenine and uracil. Okay, you notice the similarities of their structures. Between the thymine and the uracil. They're so close to each other structurally. And letter C, this is your 3 hydrogen bonds, okay, a triple bond, which is found in both DNA and RNA, which is your guanine and the cytosine base pair. In letter D, this is a more detailed zooming in structure of your double-stranded DNA. We call it dsDNA. Okay, they are consisting of what? Antiparallel strands of nucleotides held to one another by what kind of bonds? Hydrogen bonds. Okay, hydrogen bonds, and they are complementary between the bases. As you all know, A pairs with T and G pairs with C and vice versa. Note, I take a look that scientists number the carbon atoms in a nucleotide sugar component as 1' here until 5'. 1', 2', 3', 4', and 5'. Notice this ring of your carbon atoms in a nucleotide sugar components. So you can drop your answers from the comment sections below. What structures do DNA nucleotides and RNA nucleotides have in common? What do you think? Let me know. Next, we go ahead with the structure of your prokaryotic genomes. In this particular lesson, we describe the structure, the shape, the number of chromosomes in a typical prokaryotic cells, and also we will describe the structure and function of your plasmids. In letter A, you will see here a bacterial chromosomes, which are packed in a region in your cytosol. Okay, we call it the nucleoid, and they are not membrane bound. They are not surrounded by membranes. For letter B, this is a packing of a circular bacteria of the circular bacterial chromosome into loops. Okay, unlike your eukaryotic chromosome, your bacterial chromosome is in circular fashion or in circular structure. So we see here, after the cell was gently broken, they're open to release the chromosome, and this extra chromosomal DNA in the form of plasmids, look at this, is also visible. But what are plasmids? In addition to chromosomes, many prokaryotic cells contain one or more plasmids. That includes bacteria and archaeans. They are small molecules of DNA that replicate independently of the chromosome, and plasmids are usually circular, and they are on 1% to 20% of the size of a prokaryotic chromosome, and they range in size from a few thousand base pairs. Researchers have identified many types of plasmids, and sometimes they call it factors. Okay, They include the following. The first is fertility plasmids. They carry instructions for conjugation. Remember, conjugation is a process by which your bacterial cells transfer DNA to other bacterial cells. Okay? In essence, if you remember the horizontal gene transfer, this is how the mechanism is also similar to conjugation. Resistant plasmids are, we call it R. They carry genes for resistance to one or more antimicrobial drugs or heavy metals. Okay, this is for also for survival. Okay, bacteriocin plasmids, they carry genes for proteinaceous toxins. We call it bacteriocins, which kill bacterial cells of the same or similar species that lack the plasmid. Okay, for the plasmid lacking bacterial cells, they will eventually die using these toxins. We call it the bacteriocins. Virulence plasmids, they carry instructions for structures, enzymes, or to enable a bacterium to become pathogenic. So these are more likely to promote, okay, virulence microbes, virulent microbes or pathogenic microbes in essence. So now we try to look at the structure of your eukaryotic genomes. Okay, eukaryotic genomes is consisting of both your nuclear and extranuclear chromosomes. Some eukaryotic cells also contain plasmids, and we call it the nuclear chromosomes. Okay, this one is a figure of a eukaryotic nuclear chromosome packaging. So this is letter A, which are histones, okay, in blue-green. They stabilize and package your DNA to form a nucleosomes by linker DNA. They are tightly wounded and coiled using these histones. Okay, just remember your thread being securely fastened into a container or a tube. This is how it this is just to reduce the surface area that is being occupied by your chromosomes. In letter B, these nucleosomes clump to form chromatin fibers. Okay. In letter C, these chromatin fibers fold and they're fully organized into active euchromatin and heterochromatin. They inactive, they are loosely packed. If they are inactive, they are tightly packed. So in letter D, this is a this happens during the nuclear division or the mitosis. These duplicated chromatins will fully condense into a mitotic chromosome, okay, that is visible by light microscopes. Generally, this is the PR and the QR. If the nucleosomes were actually the size shown as this artist's illustration, okay, the chromosome in D would be like 20 meters or about 65 feet long. That's how long it is when you try to measure all of these strands that are tightly wounded in your euchromatin and heterochromatins. Now we talk about extranuclear chromosomes in your eukaryotes. Again, these are a part of the eukaryotic genomes. Not all of your DNA of a eukaryotic genome is contained in your nuclear chromosomes. Most eukaryotic cells have mitochondria, and plant and algae and some protozoan cells have what we call as your chloroplasts. Okay, both mitochondria and your chloroplasts contain chromosomes. Remember the endosymbiont theory, the evolution of the eukaryotic cell. This is how the chromosomes of your mitochondria are becoming circular in your eukaryotic cells, though researchers had thought that the chromosomes of your chloroplasts were also circular. So there are some recent researches that has shown that chloroplast chromosomes were linear. Okay, The extranuclear chromosomes of both the mitochondria and their chloroplasts resemble the chromosomes of your prokaryotes. And in this particular table, we will be characterizing the microbial genomes. So we have here the domains: bacteria, archaea, eukarya. For the number of chromosomes, we have for bacteria single because they are in haploid of one or more. Also with archaeans. For eukarya, with one exception, two or more, they are typically diploid. For plasmids, are they present in bacteria in some cells? Yes. Frequently more than one per cell. In archaea also seldom. In eukarya, we can see them in fungi, for algae, and your protozoans. For typical nucleic acids, for bacteria, they could either be circular or linear, double-stranded DNA. For archaea, they could be circular, double-stranded DNA. For eukarya, linear. For dsDNA in the nucleus in your chloroplast, circular DNA in your mitochondria and your plasmids. Where can you find the DNA? For bacteria, they could be inside your nucleoid of your cytoplasm and in their plasmids. For archaea, they could be found in your cytoplasm and in plasmids. In eukarya, they are located in your nucleus. Okay, we call it the nuclear chromosomes, and extranuclear chromosomes in your mitochondria, chloroplasts, and also plasmids in cytosol. Are there histones present in bacteria? No. Okay. They are associating the chromosomes into a non-histone protein, a small amount of that. In archaea, yes, and in eukarya, yes, because in nuclear chromosome, but not in extranuclear chromosome. Let's start again with your DNA replication. What DNA replication? We will describe DNA replication as a semi-conservative process. We compare and contrast the synthesis of leading and lagging strands in your DNA replication, and also we contrast bacterial DNA replication with that of your eukaryotes. Again, DNA replication is an anabolic polymerization of a process of DNA anabolic polymerization, which allows a cell to make copies of its genome. Though bacterial, archaeal, and eukaryotic cells package DNA differently, all of the three types of domains have or employ similar mechanisms for DNA replication. Okay, but the key to DNA replication is that the complementary structure of two strands, we have the adenine and guanine in one straight bond with thymine and cytosine respectively. Okay, if we take a look at the base pairs, the usual base pairs, DNA replication is a very simple concept. A cell separates into two strands, two original strands, and uses each of the template for the synthesis or the creation of a new complementary strand. And biologists will say that DNA replication is semi-conservative because each of the daughter DNA molecule is composed of one original strand and one new strand. In essence, you can see this in this semi-conservative model of DNA replication. Each of the two strands of the original molecule, shown in dark, serves as a template for the complementary strand, which is shown in lighter. So this is your DNA, and then the original strand in purple, and the new strands in dark green. So since we use this new strands and original strands for the second replication, they will produce the lighter shades for this figure. Okay, again, the semi-conservative. So we will discuss also the dual role of your triphosphate deoxyribonucleotides as a building blocks and energy sources in DNA synthesis. Remember the cell cycle? In the S phase where DNA is being synthesized, this structure here is your guanosine triphosphate deoxyribonucleotide, or the DGTP. Okay, like all of the triphosphate monomers of DNA, it is a nucleotide to which two additional phosphate groups are being attached. Okay, this is the phosphate groups 1, 2. Generally, we can see only single phosphate group for the DNA, but here there are two additional phosphate groups which were being attached. So in this particular figure, the energy required for DNA polymerization, or in layman's term, the addition of a nucleotide building blocks to a DNA strand, that's polymerization, is being carried out by each of these triphosphate nucleotide in the high energy bonds between the phosphate groups. Okay, so this is your existing DNA strand plus your triphosphate nucleotide, and we remove the diphosphate. We call it the pyrophosphate, and they're released in, and they're releasing energy which is used for the synthesis. Okay, so instead, from this shorter existing DNA strand, we put up a much longer strands using polymerization. Okay, so the question is, what is the difference between DGTP and guanosine triphosphate ribonucleotide, or the RGDP? It's very simple. The molecule DGTP contains deoxyribose from the first letter D, deoxyribose, whereas your RGTP contains ribose. Okay, these are both sugars. Next, we do have the initial process in bacterial DNA replication. Okay, your DNA replication will begin at a specific sequence of nucleotides, and we call it an origin. An enzyme, we call it the DNA helicase, is locally unzips the DNA molecules by what? By breaking the hydrogen bonds between the complementary nucleotide bases and the exposed nucleotide bases in a replication fork. We'll see it later. And other protein molecules were being used to stabilize the separated single strands so that they do not rejoin while the replication proceeds. Here, this is the initial process. Okay, initial process will be the cell removes proteins. Okay, we call these proteins as histones in eukaryotes and archaeans. The one that is tightly winded. Okay, this is the blue-green circular object here. These are chromosomal proteins. And from the DNA molecule, they're being removed, and then helicase, this green one here, will unzip the double helix. Once they unzip the double helix, they will break the hydrogen bonds between your complementary base pairs and form a replication fork. So this is your replication fork. And then the stabilizing proteins here, again, as I say, they keep these leading strand and lagging the two strands, okay, not to intertwine, or not to mingle again once they are being separated by your helicase. Okay, let's go and further the second process. This is the continuous synthesis of the leading strand. Okay, DNA synthesis always moves in a 5' to 3' direction. 5' to 3' direction. So the leading strand here, leading strand, okay, is synthesized towards the replication fork. This is the replication fork. This is the the intersection. Okay, so the leading strand is going into the 5' to 3' direction. The numbers that are shown here were the steps in the process. Okay, and again, the proofreading function of your DNA polymerase 3, this orange structure here, though the proofreading is not detailed in this particular artwork, replacing the leading strand's RNA primer with DNA is shown in the few steps. Okay, so in the first, number 1 here, an enzyme called Primase, this orange circle here, will synthesize a short RNA molecule that is complementary to the DNA strand being copied. The RNA primer in red provides a 3' hydroxyl group, and they are being required by the DNA polymerase 3. Okay, so there must be RNA primer that provides hydroxyl group before the DNA polymerase 3 can assist them. And number 2, the triphosphate deoxyribonucleotides will form hydrogen bonds with their complements in their parental strand. Okay, they add complementary nucleotides, and your adenine nucleotides will bind to thymine nucleotides, and guanine nucleotides will bind to cytosine nucleotides. Generally, that's the base pairs according to Chargaff. Using the energy in the high energy pathway, the triphosphate deoxyribonucleotides, DNA polymerase 3 covalently joins them one at a time. Look at this. One at a time to the leading strand. And DNA polymerase can do this, and they can add about 500 to 1,000 nucleotides per second to a new strand. And fourth, the DNA polymerase proofreads. And DNA polymerase proofreads as it extends. It's not shown here, but again, the proofreading function is being done by the DNA polymerase 3, and about 1 to 100,000 nucleotides is mismatched with its template. For instance, a guanine might become incorrectly paired with thymine, and DNA polymerase 3 can also recognize this error, okay, most of these errors, and removes the incorrect nucleotides before proceeding with the synthesis. And this role is known as your proofreading exonuclease function. And if you're familiar with computer, it acts like a backspace. Okay, delete. Okay, backspace key on a keyboard, removing the most recent error and continue to provide corrections to that error. And because this proofreading and exonuclease function and other repair strategies are beyond the scope of this discussion, okay, only one error remains for every 10 billion base pairs replicated, so cells makes an almost exact copy of their genome. Next, we have here the synthesis of the lagging strand. Okay, so this is the discontinuous synthesis of the lagging strand, and they are proceeding, moving away from the replication fork. This is the opposite. Okay, so if the leading strand will work at 5' to 3', this one is working with the 3' to 5'. Okay, and you'll see here actual Okazaki fragments in red are about 1,000 nucleotides long. The question is, why is DNA replication again termed semi-conservative? Again, in this image, semi-conservative refers to the fact that each of the daughter strands or daughter molecules retain one parental strand, and each has one new strand. In other words, each half new and half old. Very good. As a replication of the leading strand continues, the DNA polymerase, okay, or we call it the DNA polymerase 1 here, replaces the RNA primer with nucleotides. And note that the researchers renamed the DNA polymerase enzymes in order of their discovery, not the order of their action. So the DNA polymerase 3 from the leading strand is you don't have to confuse yourself, okay? They name it according to their discovery. For this number 5, the primase, okay, this blue-green circle here, molecule here, will synthesize RNA primers. And in contrast to its action on the leading strand, primase synthesizes multiple primers at about 1,000 to 2,000 DNA bases in this template strand. Your DNA polymerase 3 you see here joins the neighboring nucleotides and again proofreads. In contrast to the synthesis of the leading strand, however, the lagging strand is synthesized discontinuously. And these discontinuous segments are called Okazaki fragments. Okay, this is named after a Japanese scientist named Reiji Okazaki, which lived 1930 to 1975, who first identified these fragments. Each Okazaki fragments again consisting of 1,000 to 2,000 nucleotides. In number 7, you'll see here nucleotides pair up with their complements in the template. Again, adenine with thymine and cytosine with guanine. In number 8A, your DNA polymerase 1 will replace the RNA primers of Okazaki fragments as well as the RNA primer in the leading strand with DNA, and proofreads the short DNA segment it has just synthesized. Again, it also works as a proofreading. And lastly, number 9, there is a DNA ligase which will be sealing the nicks between the adjacent Okazaki fragments to form a continuous DNA strand. So this is just a preparatory process before we fully understand the central dogma of genetics. Now we take a look at the other characteristics of your bacterial DNA replication. Your DNA replication is bidirectional. That is, your DNA synthesis proceeds in both directions from the origin. In bacteria, the process of replication typically proceeds from a single origin, so it involves two sets of enzymes and two replication forks, each having a leading strand and a lagging strand. Again, DNA replication is further complicated by methylation of your daughter strands, in which this process is making an enzyme adding a methyl group to one or two bases that are part of specific nucleotide sequences. Bacteria typically have a methylate adenine bases, and they only rarely own a cytosine base. So in this particular figure, we see the bidirectionality of your DNA replication in prokaryotes. Replication begins at an origin, the green one, and proceeds in both directions to a termination sequence or a termination point in red. So this particular bacterial sample is the E. coli, Escherichia coli, which is 80 base pairs in size. Okay, these are bacterial chromosomes. They have single origin, but eukaryotic chromosomes, on the other hand, have thousands of origins. Again, bidirectionality. Now, what is methylation? Methylation plays a role in a variety of cellular processes, including the following. First, they control genetic expression. In some cases, genes that are methylated are turned off and they are not transcribed, whereas in other cases, methylated genes are turned on and are being transcribed. Okay, we take a look at the embryology part. Okay, we lost the webbed structures in our in our hands or palm during the development of the embryo. As it go further, reaching 3 months, 4 months. Okay. The next is the initiation of DNA replication. In many bacteria, methylated nucleotide sequences will play a big role in initiating DNA replication. So again, they could start methylation could start DNA replication. They could also protect against viral infections. In specific parts of your nucleotide sequence, they enable the cells to distinguish their DNA against a viral DNA. And a viral DNA the DNA of your viruses lacks methylation. Then the cells can then selectively degrade the viral DNA. And in the repair of DNA, the role of methylation in some DNA repair mechanisms will be discussed in the further lessons. So, eukaryotic DNA replicate much the same way as bacteria does. Okay, helicases, topoisomerases, they will unwind your DNA. Protein molecules will stabilize the single-stranded DNA. Your molecules of your DNA polymerase will synthesize the leading and lagging strands simultaneously. However, eukaryotic replication differs from prokaryotic replication in some of the significant ways. Eukaryotic cells uses different DNA polymerases to replicate DNA, and they have four the basic types. One, initiating replication, including the synthesis of a primer, which is the function performed by primase in your bacteria. The second type of eukaryotic DNA polymerase elongates the leading strand, and the third appears to be responsible for replicating the lagging strand. The fourth one will replicate the mitochondrial DNA, and because of the large size of your eukaryotic chromosomes, they will necessitate thousands of origins per molecule, and each generating 2 replication forks. Otherwise, the replication of your eukaryotic genomes would take days, not hours. Aside from that, because again, we compare it with bacterial origins initiating replication, and we have eukaryotic Okazaki fragments. They are shorter than those of bacteria. Okay, for bacteria, it's 500 to 1,000 nucleotides long. In your eukaryotic DNA, Okazaki fragments, they are 100 to 400 nucleotides only. And plant and animal cells methylate cytosine bases exclusively. So we compared, okay? The first topic we must consider if we are about to understand gene function, it's in the relationship between your genotype and your phenotype. Again, genotype of an organism is the actual sets of genes in your genome, and the genotype differs from a genome in that a genome also includes nucleotides that are not part of the genes, such as the nucleotide sequences that link one gene to another. So there are some accessory genes, okay, that is different from a genotype, okay, because they are actual set of genes. In phenotype, they refer to the physical features and functional traits of an organism, including characteristics such as your structures, morphology, and metabolism. All you can see the proteins that are present in the organism were the phenotypes, including behavior. Then the transfer of genetic information will be discussed in this latter part. Okay, we will state the central dogma of genetics, explain the roles of DNA and RNA in polypeptide synthesis. What's the central dogma of genetics? The cells do not convert the information coded DNA directly into proteins, but they must first make an RNA copy of the gene. Okay? In this copying process, we call it transcription. The information of DNA is copied as RNA nucleotide sequences and the RNA molecules in the ribosomes, then that they are being synthesized to polypeptides in a process we call it the translation. Okay, and to sum it up, these processes make up the central dogma of genetics. DNA is transcribed to RNA, which is translated to form a series of amino acids. We call it the polypeptides. Okay, why do cells do this? Cells must continually synthesize proteins because proteins are again required for growth, reproduction, metabolism, and also regulation. This synthesis also requires that they could actually and accurately transfer the genetic information that are contained in your DNA nucleotide sequences to the amino acid sequences of your proteins. And to use this analogy, okay, illustrating the central dogma, suppose that you are trying to understand this following message, which is a portion of the Oath of Hippocrates written in the Greek alphabet, this part. Again, if the Greek alphabet is foreign to you, you might have the Greek characters transcribed into the familiar English alphabet as the first step in understanding this message. When you see this one, in Philippines, we have alibata. You can translate alibata to what we could read. This one is a Greek using Greek alphabets. So this is diatimasi te klesiumai ep opilen kam nonton kata dunamin kay klesin emien epi de la se de kay adikizay erzit. And then you could begin the process of having the Greek words now expressed to English words or English letters. I will prescribe the treatment to the best of my ability and judgment to help the sick and never harmful or illicit purpose. So, to sum it up, to link analogy in that Hippocratic statement, to a ribosome, your DNA is like a foreign language, which is written in a foreign alphabet. Thus, a cell must continue to use the processes analogous to those just described. It must first transcribe the foreign alphabet of their DNA nucleotides, that are the genes, into a more familiar alphabet of your RNA nucleotides. So after that, then it must be translated to the message formed by these letters into words. And these words are your amino acids that make up the message, which is the longer chain, which is your polypeptide. In this way, a genotype can be expressed as a phenotype. There are a few exceptions for the central dogma. For example, some RNA viruses transcribe DNA from an RNA template, and a process we call it the reverse, we call it the reverse of cellular transcription. That's for the special RNA viruses. Now, to better visualize this figure will help you understand the central dogma of genetics. A cell, again, transcribe RNA from DNA gene and then synthesize the polypeptides by translating the codes carried by the RNA molecules. And polypeptides will identify and determine the phenotype by acting as structural, enzymatic, and regulatory proteins. Before we end, we go back to our case study about Ben. The doctor and nurses are able to stabilize Ben, and he is being transferred to the hospital. Upon arrival, Ben has a fever of 103.1 degrees Fahrenheit, and his white blood cell count is elevated. We have blood and urine sample cultures are collected and sent for lab analysis. The elevated WBC count suggests an infection, so the doctor starts Ben on meropenem, an ultra-broad spectrum antibiotic. The next day, Ben does not appear to be responding to the meropenem treatment. The lab results indicate a multi-drug resistant Klebsiella pneumoniae present in Ben's blood sample. This diagnosis is cause for concern because this organism is like a carbapenem-resistant member of your Enterobacteriaceae, a member or a family of bacteria that has a high level of resistance to multiple antibiotics. The doctor puts Ben on a course of high-dose tigecycline, an antibiotic that has shown effectiveness against carbapenem-resistant Enterobacteriaceae, or the CREs. The next few days, they bring little improvement to Ben's condition. His condition continues to decline, and he ultimately suffers from multiple organ failure and eventually dies. Guide questions. The prevalence of CRE infections in healthcare settings has increased significantly in recent years, in part because these organisms are able to share plasmid-encoded resistance genes. Which method of horizontal gene transfer is most likely to be responsible for the transfer of the plasmid between bacteria? Explain your choice. And second, Ben most likely acquired the infection while he was in the hospital recovering from his stroke. What are the possible sources of infection in the hospital that might have led to Ben's infection? Take a think of that questions, and we'll discuss that one in the comment sections below. And this is microbial genetics. This is the first part. Transcription events will be coming next. Thanks again for watching, and have a nice day.